I'm looking forward to all the nice discussions we will have later and all the questions you guys might have. So how do I change this? With this one?
Yeah.
Yes. So I can start by introducing again, like Tanjana did really well, so I will just introduce my group. I am doing my PhD at the University of Turin. My supervisor is Cristina Tuni. Our lab is reproductive behavior lab, so we are working on the reproductive behavior and its ecology and evolution on crickets and spiders. And I'm on the first year of my PhD.
And well, you already introduced, so I'm Sofia. I'm doing my second year of PhD at the University of Padua in the laboratory of Claudia Gasparini. In my laboratory also they work both on reproductive behavior. Actually I'm the only one that works on insects. The other people are working more on fish, for example guppies and zebrafish, and on birds. Now I will let Yasmin.
Yes, okay, now without further ado, let's get to the topic. Maybe it's better if I come through, you guys are there. So, the Earth is heating up, as we are all aware of. And each year we hit a new extreme temperature of global average maximum. And we, as humans who are not so temperature dependent right now, feel it. We can compare between our childhoods and now and see that the climate is changing somehow. The animals, the other animals, who are way more temperature dependent than us, are definitely feeling it. And to these changes in the climate, they either respond by adaptations, if they can't adapt, they get extinct. So one of the most common adaptations is phenology change. And in a research done, they saw that many species of birds are now laying eggs earlier in the season than they used to, and this is due to climate change. Another adaptation that could be is location change. And in this case, I want to talk about these beautiful birds called rock ptarmigans. These are birds that have adapted to very cold climates. They live on the top of the Alps, and they have this very amazing adaptation to change their feather color between winter and summer. And when the summer comes, they form this brown plumage, and in the winter, they are all white. They have the white plumage. However, now that it is snowing less and less, the ptarmigans with the white plumage are way more promiscuous for the predators and therefore get predated more. So in this case, they cannot also do a range shift because they are already living in an extreme. And they are under the risk of extinction. Another example to this could be tropical species, but we will talk about that more later. And climate change is not only the steady increase of temperature over years, but it's also the extreme events that occur way more now. So, like floodings, maybe you heard last week there were very unusual floods in Europe, and heat waves. So heat waves are a part of our lives in the temperate region. However, over years now, the frequency and the duration and also the intensity of these heat waves are increasing. And last year it was like a summer of never-ending heat waves in the US, in Europe, Asia, everywhere. The heat waves are like, it gets to the maximal temperature or a bit more than that than we are used to. And then it goes on like this for days. So in Turkey, we are used to having 41 degrees once or twice in the summer. However, last year it was 41, 41, 41 for several days, and that was really unusual. And then I read a bit about heat waves in Madagascar. So because it's a tropical climate, it was actually virtually impossible to have a heat wave. But now with the 1.2 degrees warming, it is still a minor possibility. And with the close feature of 2 degrees warming, it might also become a part of your lives in Madagascar. And as biologists, we are interested in how these changes will affect the animals. And I must say, this effects of temperature on animals was really well studied. And people usually used critical thermal limits to understand this. So critical thermal limits, CTL, is a measure that people use They basically increase the temperature until the animal faints or dies, and they call this the thermal limit of the animal. And this measure correlated well with species distributions, and it was used for climatic models or estimating range shift capacities of animals. However, now another measure is being introduced called the thermal fertility limit, and it is when the animal loses its ability to reproduce. And we know that if a population cannot reproduce anymore, it cannot sustain. And here in this plot, you can see a study done on Drosophila, and we see that temperatures that are needed to lose fertility is much lower than the critical temperatures. So we might be overestimating how species will cope with the climate change. And there are some studies done on the effects of temperature on different levels of reproduction, because reproduction is not so straightforward as sperm healthy reproduce, but it has more levels. Like in this fish, they saw that heat waves, higher temperatures for a few days affected activity and boldness, which might in return affect their ability to find mates. Temperature has effects on cognition, mating dynamics, different pathways of gamete production. So there are multiple levels. So now to summarize the problems that I introduced and finally present my research topic on the PhD, we said that non-lethal heat stress can affect population survival by affecting fitness. And climate change is not only the increase of temperature over time, but it's also the extreme events happening more and more. So my complete question is, how do heat waves affect reproduction? And I will ask this question in these insects. Grillus bimaculatus was my main plan, but now also Grillus madagascariensis is added, I can say. And why do I want to ask this big question in a system of insects? First of all, insects are extremely important for the ecosystem. They are everybody's food. They are an indicator of the ecosystem health. But also they are short-lived and ectothermic, which means that they are way more affected by the outside temperature changes. And short-lived because even a 3-day heat wave can make up a big part of an insect's life. They are well studied, easily kept in the lab. Like Grillus bimaculatus, we have its own whole genome, and people have done multiple studies on their songs and systems. So it is it's a very good system to ask novel questions like this. They perform variety of reproductive behaviors we will talk about soon. And here you see a map that I took from iNaturalist. So Grillus bimaculatus, it's called African field cricket, but also Mediterranean field cricket, because it's everywhere. You can hear its songs in all the fields in Italy, in Spain, but also I heard now they're very common in Africa. What I was very surprised and also happy about was that they're also in Madagascar. They came here somehow with human transport. But now this provides me the perfect system, because I can compare the effects of heat waves on the same species between two climates. One that has adapted to a temperate region and one that has adapted to tropical region. And it is very important to study tropical and island species and include this to our experiments, because we expect that they will be affected more by climate change. Island species because they have less genetic diversity, and tropical climates because they usually already live in extreme heats and their metabolism is super high. Yes, so we can continue. So now I would like to introduce my methods that I will use. So I did the first step now. I came to Madagascar. I'm collecting Grillus bimaculatus and also Madagascariensis here. And I will collect Grillus bimaculatus also in Italy when I go back. Then I will simulate these heat waves of... Hi! Sorry. Yes, I will simulate heat waves of different intensities and durations. And I will decide on these heat waves according to the situation, let's say. For example, here I have bimaculatus in the lab, And I read that Antananarivo usually reaches 30 degrees in summer. So I am giving now 30 degrees 3 days in a row. So it's a realistic scenario. But however, in Italy, I will do my heat wave with 39 or 40 degrees because in there that is the realistic scenario. And right now, I am doing this only on adults, and I'm expecting changes in metabolism and behavior. However, I want to also do this on subadults. I want to heat wave subadults and look at its effects on reproduction on the adult stage. Because as subadults, there is the wing development and gonad maturation, and they're very important, of course, for reproductive behavior. So I think what happened in subadult stage can adapt the reproduction in the end. And after I simulate these heat waves, I will look at the reproductive behavior, which is singing, mating, and fighting. Fertility, the sperm success, egg success, fitness, the amount of eggs that are laid and eggs that hatch, and gene expression. Because in some stages of subadult, the gene expression for wing development is the highest, for example. And the songs are a big part of the reproductive behavior. I will explain this on the next slide. So right now, what am I doing in MBC? We went to the field. These people helped us. We're so lucky to have worked with them. They found lots of crickets. So we have Immaculatus, that is the one. And we have Madagascariensis, which you are probably very used to because there's a whole farm of it there. And this is our setup. So we make videos of a control male or a heat waved male together with a control female. And we record to see how many times this male will chirp, how much time will the female take to accept him, will there be any aggressive interactions, and we also have a microphone inside to check for the song. Yes, that's pretty much it. And finally, my predictions. So, I expect that females will choose the heat waved ones less because they will have effects on sperm. And females are very selective and very good at choosing the right male. Because, yes, there are studies on it. Females can actually recognize which male is bigger from the song and go to him. Or even when they stabilize the size, the male she chooses to mate ends up having the most successful sperm or immunity. So we don't know exactly how this is happening, but females can get a lot of information from the song. So we will analyze the songs that we record. And a study that was made by my supervisor last year, you see these are from Italy, so 39 degrees was the heat wave. They saw that the singing duration increased on the heat waved adults. And increase means it is less successful because in a perfect mating scenario in the lab, the male sings just a few times and the female is like, okay, you're good, and then they mate. However, in this case, sometimes males sing and sing and sing, and the female just doesn't accept it. So that was the case with the heat wave population. Yes, after I am done with heat waving the males, I am also planning to do the same experiment with females, and in this case, I also expect the egg quality to lower, the fitness to lower, and in my comparisons between Madagascariensis and Immaculatus, I am actually very curious what's going to happen in that case because there are really no studies on Madagascariensis, so whatever we see will be new. But I expect lower thermal plasticity in Madagascariensis because it is native to here and it is adapted to evolve to this tropical climate. However, Immaculatus, even though it's adapted to here now, maybe it has some genetic residuals and has more thermal tolerance. And with the same logic, I also expect the Madagascar population of Immaculatus to be affected more than the Italian population. So, I hope I didn't tire you so much, and thank you so much for listening. Now, Sofia will talk about the hissing cockroach.
Now we'll talk about my PhD project that is about the impact of climate change on fertility and behavior of the Madagascar sea cockroach. So first of all, I wanted to tell you why I choose the Madagascar sea cockroach. So the cockroach is usually like seen as a pest, but actually what I wanted to tell you is that the most percentage of cockroach species are actually not pests, just like a small percentage are human pests. But why did I choose this animal? First, as also Yasmin explained before, it's an ectotherm organism. That means that they're not able to control the temperature as endotherms do, and so the climate change can affect them more. Second, this cockroach is really interesting from a reproductive point of view because it's viviparous. That means that it doesn't lay eggs like other insects do, but it keeps the eggs inside the body in a ootheca, and then it's like giving birth to the young individuals that are actually similar to the adults but small. And third, it's really easy to rear. Actually, I'm doing the experiments also in Padova, and in Italy we can find these cockroach even if they are endemic from Madagascar because they sell them like in pet shop, and actually some people have them like in their house as pet. And but usually they sell them like to feed other animals in reality. They are also very interesting because there is a high sexual selection and a dimorphism between male and female. The last reason is actually also for the grillos because it's a tropical insect, so the effect of climate change on them can be more risky because they are used to higher temperature. And also, even if they can be the most vulnerable insects, there are really lack of studies on this area.
Sofia? I'm sorry, the dimorphism, is it like male and female or it's just another bug?
No, this one is young. This one is a male, and this species doesn't have big horns actually, but you recognize usually the male because they have horns that actually used to attract the female. So first of all, I will speak about the project that I'm doing in Padova. So the study in the laboratory. I'm doing them in the Esapolis Museum, that is a museum of arthropod in Padova. And here you can see my laboratory. We keep the cockroach at a temperature of 28 degrees and a humidity of 16% more or less. And you can see that they are like red light because they are nocturnal insects. So we inverted the light-dark cycle so that I don't have to work like during the night, but I can study them during the day. This project I'm doing on the heat wave, I'm not doing it alone, so I'm doing it with these 3 students. Veronica and Michele, that are master students. They choose the picture, I didn't choose them. It's their responsibility. And Max, that is a bachelor student, and actually they are still doing this experiment while I'm here in Madagascar. So my aim is to study the effects of the heat wave on the behavior, on the survival, and on the reproduction. So I wanted to study both the effects on survival and reproduction because from studies we saw that reproductive traits can be more affected by heat wave. So maybe a temperature that don't kill the animal can make them sterile. And of course, even if an animal and the insect is alive but it cannot reproduce, this can lead the species to extinction at the end. So this is a timeline of my experiment to explain you how it's planned. There is a pre-treatment and a post-treatment that is more or less specular. So I will do the same experiments before and after the treatment of C-stress, both with the control and the heat-stressed individual. Since now, we tested 10 groups that are composed of 6 individuals each, 5 control and 5 treatment. My plan is to increase still the sample size once I will go back to Italy. So now we'll explain you the pre-treatment experiment. So we performed some behavioral tests that are used as proxy of the condition of the cockroach. So to see the condition before and after the heat wave. First of all, we did an exploration and boldness test that is used first to see like how a cockroach can be shy or bold, so the personality of cockroach, and also to study their exploration. So their activity, the speed of cockroach in the arena, the distance, and also if they stay more in the border of the arena or in the center. Then we explore the aggressiveness of cockroach, in particular of the males, because they fight also reproductive behavior to attract the female. And the writing of the cockroach to see actually their condition, and the writing is the ability to turn themselves if they are turned on the back. This is a behavior that they need to use because it actually happens very often during the fighting that they are turned into their back. In addition to the behavioral test, we also did the cuticular hydrocarbons analysis. First of all, I don't know if you know what cuticular hydrocarbons are. Cuticular hydrocarbons are these chemical compounds that are present on the cuticle of insects, and they have different functions. Between them, there is a waterproof function that helps insects to protect themselves, for example, from desiccation, but also a communication function. We want to focus more on the communication function because these insects are social, and so of course communication in a social group is really important. And what we want to investigate is if heat wave can actually affect these compounds, so both from a qualitative and quantitative point of view. And so if maybe the communication between the individual can be affected, and as a consequence, for example, also the reproduction of these insects. To do this, we just use a wipe on the pronote with an exam washed filter paper sheet. Then I will send the sample to Federico Cappa, that is a researcher of the University of Firenze I'm collaborating with, and he will do the extraction, and then he will put the sample in the gas chromatography. In this way, we will obtain the peaks analysis that will help us to understand the quantitative and the qualitative results of the chemical compounds. In addition, we also studied the hierarchy of this cockroach. As I said before, they are social insects, and if some individuals that didn't know each other are placing in the same arena, the hierarchy has to be set up. So we place 3 female and 3 male that didn't know each other in our arena, and then we observe them for 3 days, and we keep on taking picture with a photo trap for 5 days. Actually, in that way, we could observe the dominance inside the group, because usually the dominant male goes in a elevation. Actually, here you can see there is like an elevation point in the arena, and so analyzing the picture, we could observe the dominance inside the hierarchy. We wanted to do these experiments because actually the males that are dominant can be the most affected by the heat wave, because it can be that they use all the energetic resources to allocate more in the aggressiveness and in the mating, because the dominant male are the one that can mate more than the other. Then, after the heat stress experiment, we did the treatment. So we did 3 days heat stress, and we simulated the heat wave using an incubator. Actually, these are eggs incubator, like for the chicken. And we decided to use 28 as control group because it's also the temperature we take the cockroach inside the laboratory. And then 38, that is so the treatment temperature, and we choose this temperature by testing actually different temperature with few individuals, and also by checking the temperature that a heat wave can actually occur. And we decided to do the experiment with a constant humidity because we wanted before to see how the temperature affects the behavior and reproduction without inserting also another parameter. After the treatment, so we did the post-treatment experiment. So we repeated the test that we did before. We also repeated the cuticular hydrocarbon analysis, both right after the heat wave, but also 1 week after, because we wanted to see if the effects were just temporary or long-term effects. And then we also added 2 more behavioral experiments. One is the thermal preference. So they could choose between a part of arena that was colder and one that was warmer, to see if the cockroach that were already used to a warm temperature could choose the cold temperature because they knew the effects of the warm temperature, or the opposite. And then we wanted to see the response to stimulus. because actually if a cockroach cannot respond to a stimulus, that can be risky for it, because they can, for example, they cannot escape from a predator. And what about the reproduction? Actually, as I said before, it's really important also to study the effects on reproduction. We still have to do the experiment on them, more for a logistic point of view, because to study the sperm production and vitality, I have to sacrifice the cockroach, because we have to dissect them and take the testes. But of course, if I wanted to do behavioral tests on them, I couldn't. And so I'm planning to do this when then I will go back. And we're planning to study the sperm production and vitality, so to see the quantity of sperm that is produced before and after heat wave, and the vitality of the sperm. And also we want to study the effects of the offspring of cockroach. This is actually a more long study if you compare it with other insects, because the cockroach takes 3 months of gestation to give birth to the offspring, and then the offspring to become adult takes other 6 months. So here in this picture are actually the sperm not of the cockroach but of wasp, but it's actually the same form of the cockroach sperm. And what do we expect from this experiment? So we expect first unimpaired body conditions, so that is translated in a higher time of writing of the insects, a decrease on the activity level, so we will study the tracking of the cockroach in the arena. And then we expect a change in the cuticular hydrocarbon composition that can move more from compounds that are used for communicate to compounds that are more used to protect themselves from desiccation, and a decrease of sperm vitality production. Studying the dominance of male, we can also try to understand if there is a trade-off between the dominance and the tolerance to estrus, because they allocate more energy in mating and aggressiveness. But actually, some studies show the opposite result. So some studies show that the dominant male can have a better genotype and so can be less vulnerable to the heat wave. Okay, so this was my work that I'm doing in Padua, and now we present the field work in Madagascar. That is like the reason why I'm here actually, and why we did this study. So first of all, I did this study with Yasmin. And that is like in all the pictures, so you can see her. And we decided to do these studies because actually we don't have studies of these species in their natural habitats, and I think it's really interesting to study insects not just in the laboratory but also in the field, because this can help us to know more about the species and also to know more or how to study them than in the laboratory. For example, in the laboratory and in the literature, when they describe groups, they always describe group composed by half male and half female. But actually in the field, we found colonies with a lot of female and just one male. So this is really like different from what was described before. And then we want to study, because actually, yes, they are tropical species, so they are more vulnerable. And according to the World Meteorological Organization, Madagascar is considered as the third ranking country in the world that is most vulnerable to climate change. That is because, as you I think know also more than me, there are a lot of endemic species here, a lot of biodiversity, the weather is so different in every part of Madagascar, and so the climate change can take a lot of effects in this country. So when we did our study in Kirindi forest, so a dry forest. We were staying at the German Primate Center, that is the actual center that studies more lemur. And we did our study at the beginning of the dry season, so from the beginning of April to the end of May. Our plan was before to do a life history study, actually before, because there are no study on the species. So our first step was to sample the cockroach, and this was like the most challenging part actually, because well, first they are nocturnal insects, so we had to go to the field in the night, but the problem is that their favorite habitat are the dead wood, and they know how to hide really well in this wood. So when the wood was really, really dead, we could cut it, and also like we can say here, we took it to the center, we tried to open it, and we found the cockroach inside. But sometimes like this one was still a really, really good tree, so they just hide in these holes and then you could not take them. So sometimes we observed them, but then it was really difficult to collect them. So we tried with a lot of strategy. Like here we tried to put a glue trap that was actually made for cockroach, like for the cockroach we have in the house. And it actually worked, because here with the photo trap, we see that the cockroach actually went on the trap. They ate the banana that we placed on it, but then they didn't stick, so then they went away. And then we tried with other trap, like with a water bottle, with a vaseline, so they could not escape, and it actually worked. But it was the only night that it was raining, so we collect the cockroach dead inside the water. So we tried a lot of strategy, but at the end we could collect 104 cockroach. And every time we collect them or we save them, we also recorded the ecological and environmental data of microhabitat, like GPS coordinates, temperature, humidity, to understand more actually about the species. When we found the colonies, because actually we found 2 different species, and I don't know the name of the species because we still have to identify, so I won't say nothing because I don't want to say the wrong species. But we found 2 different species. One was staying more alone, so we never found them in the colony, and we also found them also more close to urban building. So, for example, Yasmin found one in her mosquito net while going sleeping, while the other were almost all the time in colony, and they were more into the wood or on the leaves. When we found the colony, we also took the features of the colony, so the colony size, how many male and female were inside the colonies, if they were whole adult or just young, the weight, the picture of all the individual. Our second aim was then to study the role of the social group in the stress tolerance, as we found some of them that were in colony and some of them that were individual. So we divided them into treatment, one that was social group and the other isolated. And we studied again the effects on the survival and on proxy of health conditions, writing, exploration and boldness. So the same experiment that I actually did also in Padova. And also here we wanted to analyze the cuticular hydrocarbon. To simulate the heat wave, there we didn't have an incubator, so it was actually more difficult, and we use heat maps that work well, but of course it's more difficult to keep the right temperature, like with incubator. We decided so to increase the temperature following the temperate day excursion, because actually we measured for 1 month the temperature in Kirindi, and we observed a very high excursion during the day and the night, especially on the temperature. And so we decided to increase the temperature following this, because some studies also show that an increase in temperature and excursion temperature can actually affect the animal behavior in a different way than just using a constant heat wave. And we increased the temperature about 10 degrees, but it was 10 plus minus 3 because of course it was more difficult to control the temperature. After the test on the adults, we also did some preliminary tests on the young individual. We could just do preliminary tests because we had the young individual just when the female pregnant gave birth to the young individual. And so we had just like the last week to test them. But we could have at least an idea on how the heat wave can affect the young individual, because from other studies on insects, they can be more vulnerable than adults. And in this case, we test the writing, exploration behavior, and we did just a really short stress of 1 hour and a half, because after 1 hour and a half, I already observed that a lot of them were died. And also in this case, we tried to split them, some in colony and some isolated, to see if there were differences. We also observed the social behavior. So we placed again the photo trap, and we took picture to see if the cockroaches stay together or alone before, during, and after the heat wave. This was actually also planned to do with the adults, but the problem was like a logistic problem, because we had to remove the lids of the boxes, and Gecko would come to eat our cockroach, so we gave up with this try. What do we expect in this case. First of all, we already saw that the survival was different between the colony and the isolated, because in the colony there was no case of dead cockroach because of heatwave, while with the isolated there were actually some dead cockroach, both in the adult and in the young individual. As Tangiona said before, we also still have to get the results of all these tests. But what we expect also here is that the social group are so less affected by heatwave than the isolated individual, because they can act more social behavior to protect themselves. Also here we expect a decrease on the activity level and on the body condition. We expect a higher vulnerability in the young individual than in the adults. And we also expect to see a change in the cuticular intercargo composition, both because of heatwave, but we also want to investigate if there is difference between the wild population and the laboratory population, because actually the laboratory population is in captivity since a lot of generation. I actually don't know who collected them the first time, so we want to know if there is actually a difference in this. So that was all. Thank you for your attention, and thank you also Tangiona for organizing the seminar. I hope you have maybe question or curiosity to both me and Yasmin.